Salam and hi, today I will be presenting a project on the extraction and comparison of caffeine content in green tea and black tea. Caffeine is a naturally occurring substance found in tea leaves, coffee beans, and even cocoa beans, which are used to make chocolate. It's also commonly added to soft drink and energy beverage. At room temperature, caffeine appears as a white solid, and it belongs to a group compounds called alkaloid. These are nitrogen-containing substances derived from plants that often have a noticeable effect on the human body. Caffeine acts as a stimulant, increase alertness and energy. It is mildly habit-forming, and stopping caffeine intake suddenly can lead to withdrawal symptoms like headache and irritability. Although there's no confirmed scientific proof linking caffeine directly to cancer or heart disease, animal studies suggest it may act as a mild thyrotogen, meaning it could potentially cause birth defect. Because of this, pregnant individuals are advised to limit their caffeine intake. Here are a few key properties of caffeine. Scientific name 137-trimethylxanthine, molecular formula C8H10N4O2, molecular mass 194.19 gram per mole, and melting point 238 degrees Celsius. In this project, we use extraction to isolate caffeine from tea. Extraction is a separating technique that relies on the different solubility of substances in a specific solvent. It's especially useful when the target compound, in our case caffeine, is mixed with other substances that act as impurities. The mixture, also known as a matrix, can be either solid or liquid. We treat it carefully, select the solvent by preferentially dissolve the target compound. Ideally, this solvent should be easy to evaporate and chemically unreactive. When the mix is solid, like tea leaf, the process is called solid liquid extraction. A familiar example of this simply brewing tea, where hot water acts as a solvent that extracts a mix of substances from the tea leaf, including caffeine, tannins, and polyphenol. The objective of this experiment were to analyze the total ratio of caffeine present in caffeinated drink, and second one, to compare the caffeine content of green tea and black tea. The problem statement we explored was which caffeinated drink, green tea or black tea, contained a higher amount of caffeine. 15 gram black tea was weighed and 100 ml of distilled water was added. The black tea was boiled around 80 to 100 degrees Celsius. The black tea was filtered 3 times to separate the unused material. Then the raw black tea was transferred into the separating funnel. 20 ml of dichloromethane was added. The separating funnel was shaken gently and opened up the opening every 5 seconds to release build-up pressure. Flow out the dichloromethane solution into the conical flask. Dichloromethane is then boiled to evaporate the solution, and the precipitate was scraped out into the pieces. The whole process is repeated using green tea. After performing the extraction and measuring the caffeine content, we found a clear differences between black tea and green tea. Black tea yield 0.0282 gram of crude caffeine, which is about 0.188% of the original tea sample. For the green tea, yield only 0.0031 gram, around 0.02% of its sample. This result confirmed that black tea contains significantly more caffeine than green tea, making it strong stimulant. The dichloromethane extraction method we used was effective in isolating caffeine from both types of tea. For the discussion, caffeine is the most widely used psychoactive substance found in coffee, tea, and soft drink. It is polar and soluble in water. Caffeine contains three functional groups: amine, amide, and alkyne. It behaves like an organic amine base. Extraction process. Boiling water was used to steep tea leaf because high temperature increased caffeine solubility. After extraction into hot water, dichloromethyl was added as the organic solvent. Dichloromethyl is a volatile, colorless liquid with a slightly sweet smell. It is immiscible with water and can dissolve organic compounds like caffeine. The solution was cooled first before adding dichloromethyl because dichloromethyl boils at 40 degrees Celsius and may evaporate if added to hot liquid. A separating funnel was used to form two layers: bottom layer which is organic, dichloromethyl and caffeine, and the second one, the top layer, which is aqueous water. The funnel was gently swirled to avoid immersion. Tannins from tea also dissolve in hot water and may mix with caffeine, affecting purity. The organic layer was evaporated to isolate crude caffeine. Why green tea has a lower percentage yield than black tea? First, lower initial caffeine content in green tea. Green tea naturally contains less caffeine per gram of dry leaf compared to black tea. Even if caffeine is extracted in grams, when compared to the initial mass use, the percentage yield will be lower. Second, stronger interference from tannins. Green tea is rich in tannins, which can bind with caffeine, reducing its extractability into dichloromethyl. This lowers the efficiency of caffeine separation, even though some is still extracted. Third, less leaf breakdown in processing. Green tea is minimally processed, meaning the leaf cells stay more intact. This makes it harder for caffeine to diffuse out during boiling, resulting in less efficient extracting relative to the total caffeine present. Fourth, possible experimental loss of impurities. The crude caffeine extracted may include impurities, which is tannins, especially in green tea. This means the actual amount of pure caffeine may be even lower than measured, lowering the true yield percentage. And the last one, incorrect assumption of initial caffeine content. If the percentage yield is calculated based on the expected caffeine content in black tea and the same time is applied to green tea, it skews the yield lower because green tea simply has less total caffeine to begin with. Innovation inspired by this project, I would like to propose a mobile app called Caffeine Tally. Caffeine Tally is designed to help users track their caffeine intake from tea and other beverage. By scanning a product barcode or selecting the type of tea, user can instantly see an estimated caffeine amount based on data gathered in this project. Features of the app include a daily caffeine tracker, personalized health tips, especially for sensitive group like pregnant individual or teenager, brew time adjustment tips, and a tool to compare different tea types. To enhance user experience, tea brands could include QR codes on their packaging, linking directly to the app for brewing guidance and caffeine information. This app turns scientific data into a practical health tool for better lifestyle management. Conclusion: through this project, the extraction and analysis of caffeine content from green tea and black tea showed that black tea contains significantly more caffeine than green tea. Specifically, black tea yields 0.0282 gram of crude caffeine, about 0.188% of the sample, while green tea yields only 0.0031 gram, about 0.02% of the sample. This demonstrate that black tea has a higher caffeine content, making it a stronger stimulant compared to green tea. The extraction method using dichloromethyl was effective in isolating caffeine from both tea types.